Okay, so the next speaker is Vernon Dunn. Vernon's started his third year in a PhD program in biological sciences here. And I'm going to stop talking about him and let him get up here.
Okay. Good morning, everyone. I'm going to talk a little bit about myself and about my research, and hopefully bore you guys to sleep because it's pretty technical stuff. So let's jump right in. Just an overview of my talk. It looks like it'll be very long, but it won't be. Hopefully, I'll be nervous enough to talk through it. So I'm going to start with a little bit about me. Born and raised in New Orleans, I attended Metairie Park Country Day High School in Old Metairie. Any of you are familiar with that little private school in Old Metairie. I attended Xavier for my undergrad degree. My bachelor's is in psychology, pre-med, because once upon a time I thought I wanted to go to med school, and then that changed. After I graduated, I worked, before I graduated, in about 2 years after I graduated, I worked at Ochsner Medical Facility in New Orleans on the inpatient psychiatric unit. So a lot of acute psychiatric disorders, because again, I wanted to be a medical doctor. And I also worked in an office similar to Fogier's Victoria's position at Xavier. So I was working on the inpatient psychiatric unit, and I was also helping undergraduates find graduate school programs, specifically PhD programs. So actually, I'll tell you a quick story about how I ended up here, because like I said, I wanted to go to graduate school. I actually brought a group of students to LSU to visit the law school. And while I was here, my boss from Xavier introduced me to the guy who was in Ms. Torrey's position before her. So we started talking about schools, and he said, "Oh, well, you should look at getting a master's degree, just in between." Blah blah blah. I ended up looking at professors, and I found my professor. She's here, and actually, I don't think she knows the story. So I found my professor, and I sent her an email and said, "I'm interested in working in your lab. Do you have any spots available?" Actually, her husband, who is the associate chair of the department, got the email and said, "Oh, well, this made it to me, but I'll put in a good word for you," kind of thing. So I was invited up. I sent in my application. I was invited up for school, and the way the recruitment works at LSU is that you meet with professors in the department throughout the day for about 10, 15 minutes, and then you meet with, so you can decide where you want to do your research. So I met with everybody for about 15, 20 minutes, and then I met with Dr. Gleason, and we talked for about an hour and a half, 2 hours, not really about school, but just about life. So I like to tell that story because it's very important if you're interested in graduate school to find a professor that you can connect with, that you have, that you feel can really motivate you and push you to do well in your program, because graduate school is very stressful, and it can be a very lonely kind of road because you're spending a lot of time in the lab by yourself doing things. So it's very important to, when you're searching for graduate schools, to make a connection with the professor at a school in a department to kind of help you to save your life and help you make it through graduate school. So now for the boring stuff. So I studied synaptic transmission in the retina. Just to give an overview, this is obviously a picture of the eye, and in the back of the eye is the neural tissue, the retina, which is involved in processing visual information. So zooming in a little bit on the retina. You guys may be familiar with rods and cones, which are involved in detecting photons and processing that signal and converting it into an electrical signal that is eventually sent to the ganglion cells and to the bipolar cells into the brain. But specifically, in the inner retina, amacrine cells are one set of interneurons that are involved in mediating inhibitory interactions, the inhibitory interactions and signals in the retina. So amacrine cells are the focus of we study in our lab. So why endocrine cells? There are many reasons why. There are many different types of endocrine cells. There are many endocrine cells in a given retina. But for example, this is an image of starburst endocrine cells. It's recently it was discovered not too long ago that endocrine cells are involved in detecting motion. So if you see like this light moving back and forth in your eye, endocrine cells are one of the main cells that are detecting that motion and sending the signal to your brain to understand what you're seeing is happening. I probably started that. A little bit about my technique. This is where it starts to get real technical. So we harvest our endocrine cells from chicken embryos. So we get chicken eggs, and after about 8 days in the incubator, we do a culture procedure to isolate the endocrine cells in little petri dishes. So after about 8 more days in culture, the endocrine cells form synapses with each other, and these synapses have the same physiological activity as endocrine cells in the intact retina. So what we do is called electrophysiology, where we can use a patch pipette to clamp onto a cell, these very tiny cells, and we can either record the voltage or the current associated with the physiological activity going on in that cell. A little bit more about our lab. Again, we study synapse transmission in retinal endocrine cells. Specifically, we're studying the effects of nitric oxide signaling. These endocrine cells form GABAergic synapses. I'm not sure if you're familiar with GABA. It's the major inhibitory neurotransmitter in the body. So these GABAergic synapses are ligand-gated chloride channels. So we're really trying to understand the function of these synapses. A little bit about nitric oxide. It's synthesized in arginine by nitric oxide synthase. More relative to our cultures is neuronal NOS, is the isoform, the calcium-dependent form of nitric oxide synthase. Nitric oxide has many different functions in the retina, such as modulating light responses in the photoreceptors and cones and rods and cones. But again, we're focusing on nitric oxide signaling in the retina. So a little bit about why we're looking at nitric oxide in the retina. This is a confocal image of the different layers of the retina. The photoreceptors up here, the outer plexiform layer, internuclear layer. It's a DAF image, so DAF fluorescence when nitric oxide is present. So as you can see, nitric oxide is produced endogenously in the retina, so it's beneficial for us to study what it's doing. One of the major backgrounds about my project is that my lab has identified a mechanism that involves the nitric oxide dependent release of chloride from some internal store into the cytosol. So this blue will be the cell, and this will be an internal compartments of the cell. So when nitric oxide is present, chloride is released from some internal store into the cytosol, and this also results in a temporary decrease in cytosolic pH. A little bit more about recording and why this matters. So as you can see, there are 2 types of recordings we can do. We can use one pipette and patch onto a cell to record what will be postsynaptically, what's going on with the voltage or the current, or we can do a pair recording where we can patch onto a cell and record what's going on postsynaptically from the adjacent cell. So we patch onto the cell and depolarize. I mean, I'm probably going to use all kind of terms that just sound foreign, but I'm just trying to keep it as general as possible. Depolarize the cell, activate voltage-gated calcium channels. This will stimulate neurotransmitter release, so GABA in these cells. GABA will bind to GABA receptors and activate the ligand-gated chloride channels. So we're able to measure the concentration of chloride or the measure of chloride on the postsynaptic side. So why does that even matter? Well, normally in this situation, if you depolarize the presynaptic cell and measure postsynaptically, you would see, because GABA is an inhibitory transmitter, you would see a small inhibitory response. However, in the presence of nitric oxide, that small inhibitory response, that small depolarizing response turns into a hyperpolarizing response. So this means that we're converting a normally inhibitory synapse into an excitatory synapse. So that's pretty cool, pretty interesting for understanding synaptic transmission. So that was some background boring stuff. Now here's some really technical stuff. Again, nitric oxide releases chloride from some internal store. My objective is to identify the cellular source of where that chloride is coming from inside the cell, into the cytosol. Endosomes are strong candidates for being this source of nitric oxide releasable chloride. They have millimolar concentrations of chloride and acidic pH, so it's beneficial for us to study, to investigate them. And the first part of my project is investigating a subpopulation of endosomes, synaptic vesicles. A little bit more why this would be important. So, amacrine cell synapses participate in serial synapses, so one-way synapse, as well as reciprocal synapses. So, in a normal situation, a amacrine cell synapse would be mutually inhibitory because of the GABA neurotransmitter. However, after nitric oxide is present, that we know that chloride is being released from some internal store, so that will cause the adjacent synapse, which is normally inhibitory, to become excitatory. So this can have very strong effects on the postsynaptic side and allow us to understand it better. I chopped this down a lot because it gets very technical. My experimental design involves a bunch of experiments, the two main approaches. First, I want to try to deplete synaptic vesicles, deplete chloride from the synaptic vesicles. And the second approach involves depleting synaptic vesicles themselves, with the idea that both of these approaches, if synaptic vesicles are restored, then both of these approaches will yield the same result. Again, so the idea is the question I want to answer is, how does the depletion of chloride in vesicles affect the nitric oxide dependent release of chloride? A lot of my initial experiments involved manipulating the synaptic vesicle cycle. This is a very simplified diagram. Again, you activate voltage-gated calcium channels. Calcium comes into the cell. Synaptic vesicles bind to the membrane and release the neurotransmitter. So the first thing was to try to manipulate or activate exocytosis and endocytosis to get synaptic vesicles to enter their content. I have a little bit of my data here. I developed a voltage protocol that would consistently activate these voltage-gated calcium channels because they're very sensitive to inactivation. So I had to develop a protocol that was repetitive enough and short enough to consistently activate this calcium current. So this is about 10 different steps of consistently activating that calcium current. It probably just looks like artwork to you, but just know that it does what it's supposed to do. And then this was more of a cooler experiment, but it was very complicated. So I wanted to verify that I'm actually stimulating the synaptic vesicle population. So I have data that shows that the calcium channel is activated, but how do I know that synaptic vesicles are actually moving? So here's a picture of an amacrine cell, and that's the electrode attached onto the cell. So then I would use this lipophilic dye, FM143 and FM210. So I don't have a diagram here, but the way that FM dye works is that you wash it into this, you introduce it into the petri dish, and it gets embedded into the membrane of the cell. And as synaptic vesicles go through their cycle in exocytose and endocytose, the dye gets trapped within the vesicles and therefore within wherever they're trapped. So after my voltage protocol, you can see these aren't individual synaptic vesicles, these are populations of synaptic vesicles. And you see over here at the synapse, which this would be a synapse from another cell, where we expect synaptic vesicle activity to be happening. So we're pretty comfortable that my voltage protocol was successfully activating the synaptic vesicle cycle where we wanted to. So a little bit about my big experiment, which is very complicated, so I'm going to make it very simple, or as simple as I can. It involves activating the GABA receptors on the cell and measuring the concentration of chloride to see if we actually see if our protocol actually depletes the chloride from the internal store. So I would patch onto a cell, apply this voltage ramp protocol to activate those receptors and inject nitric oxide. And then I will measure what's called the equilibrium potential for chloride and then compare it to after I use my voltage protocol and do the same experiment again. That was like a super chopped up version. I'm sure that makes sense. So normally the data I will be looking for is that this line represents where the line crosses the zero axis is where it's showing the concentration of chloride within the cell. So after nitric oxide, this line shifts in the positive direction, which means that cytosolic chloride has increased. So some internal store has released chloride into the cytosol. So the idea is that if synaptic vesicles are the source of releasable chloride, then either that shift would be smaller or bigger, or we really don't know what would happen, but we're trying to compare what's going to happen after that experiment. So, just to summarize, nitric oxide causes a release of chloride into the internal store. From an internal store, I'm investigating synaptic vesicles as the source of this store. Another source that I'm going to try to investigate is mitochondria is being released of this chloride. So I want to thank my advisor, Ivana, my PhD committee, and then these are people in my lab. That's it.
He's got time for one question.
Okay. They're like, what?
You said so you're deleting chloride ions from the white so you said there were two papers or something deleting chloride ions from something.
One was depleting endosomes of chloride. Another was depleting the endosomes of everything. So I didn't go into detail about that other experiment. So that's going to involve endocytosis and endocytotic inhibitor called dinosaur. So it would, with synaptic vesicles, endocytosis and dinosaur blocks them from closing. So that would theoretically cause them to empty out their content by it's going to be I'm comparing two things in one experiment and then the whole experiment to another experiment.
What's the two things result in the acids I mean, I know you
yeah, or specifically synaptic vesicles.
I mean, I know you can do that, but does depleting the chloride ions, does it in any way help like
That's what facilitate
delete
So does depleting the chloride ions facilitate the process of well, theoretically, there are two different things. So synaptic vesicles have more than just chloride. They have all kind of ions inside of them. I'm just depleting one approach. I'm just depleting the chloride theoretically. And another approach, I'm going to deplete them as a whole. And I forgot to mention that the nitric oxide effect is very transient, very fast. So it occurs in a couple seconds, it only lasts about a minute. So the natural mechanisms of the cell to reload themselves happen very fast, so it's a very quick. I don't know.
Let's finish this one later. We got another speaker. So
Okay.
let me stop that there.